Welcome everybody, and thanks for tuning in to BIMS TV. My name is Colin Howe, and today we're going to take a bite out of coral microbiology. Now before I get into what my coral microbiology is, I thought it would be helpful to start by defining what is the microbiome. The microbiome is a community of microorganisms that like to live in a particular habitat. These microorganisms include things like bacteria, fungi, and yes, even viruses, but don't be too alarmed. Now, when we think of the term environment, we can think of a coral animal itself. Inside the coral colony contains a unique assemblage of microorganisms that not only like but thrive within the tissue of the coral colony itself. Now, if we took a look at and tried to describe the members of the coral holobiont, and the holobiont just means the coral animal itself plus its microbiome, what do you think we would find if we took a little tissue biopsy and we expanded and looked at all the life that's found in it? We would find, of course, coral tissue cells, but we would also find really small, ancient things like bacteria, viruses, and archaea. Now, these groups are collectively considered prokaryotic organisms, and they can outnumber the coral host cell 1,000,000 to 1. But then we can also find more complex double-bound organelle type cells, things like fungi and even microscopic algae, or in this case, zooxanthellae. And we all know that zooxanthellae are an incredibly famous part of the coral microbiome member when we talk about coral algal symbiosis. Indeed, the coral-algal symbiotic relationship continues to be one of the most well-studied and documented examples of symbiosis today. And, a lot of attention goes to the symbiotic dinoflagellates within the microbiome, not so much of some of the other species, although we're going to change that. But today, what I wanted to talk about is just some of the really interesting, significant contributions by prominent scientists in the field of marine microbiology, symbiosis, and evolution. In fact, you yourself may consider yourself a novice coral microbiologist if you've learned anything about this symbiotic relationship between corals and their microscopic algae. Now, when we hear about these groups of microorganisms, you often hear the term dinoflagellates. And yes, the type of macroalgae that is associated with coral are collectively called dinoflagellates. But another term you may have heard is zooxanthellae. And zooxanthellae simply means dinoflagellates that form a symbiotic relationship with a host. And interestingly, the family of symbionts, this is called Symbiodinaceae, which are a type of zooxanthellae that form a symbiotic relationship with corals, also form a symbiotic relationship with other phyla, including mollusca, which in this case are the vibrant and beautiful giant clams found in the Pacific. But then we can also find symbiotic relationships of Symbiodinaceae in porifera, or giant sponges. And so it's important to remember that Symbiodinaceae doesn't just form a symbiotic relationship with corals. However, I am a coral biologist, so we're going to focus on that. And this interesting paper is one of the most recent revisions to trying to describe and to categorize the countless species found within the family of Symbiodinaceae. This comprehensive and collaborative paper were actually able to describe about half of the genera found within this family of Symbiodinaceae. There's about 11 or 12 of them, and they found about 6 or 7, which also shows you how much work it goes into describing these communities, and also the rich opportunities for any of you future coral microbiologists out there who want to try to understand more about these communities and their relationship with their coral hosts. One, I can talk about, a lot of opportunities to talk about the thousands of different species found within Symbiodinaceae. But the one clade and the one species that I would like to bring your attention to is found within the Durstinium clade under this, and the species was recently named in this paper is called Durstinium trenchi. Now, the Durstinium clade as a whole in this paper is described as an extremophile, something that is found around the world and is uniquely heat tolerant. And this has its own ecological and physiological implications for coral algal symbiosis. And one of the more recently described species within this clade is called Durstinium trenchi. And the reason it was called this, it was actually named after one of the most prominent black marine biologists by the name of Dr. Robert K. Trench. And he is considered one of the founding fathers of marine symbiosis, initially studying symbiosis with bivalves and other marine organisms, but literally paved the way for us to make huge contributions in the field of marine evolution and symbiosis. A little interesting fact, the lead author of this pivotal paper, Dr. Todd Legioness, was once a graduate student of Dr. Trench's. And they dedicated this profound paper to his legacy, stating, "This paper is dedicated to to Professor Robert K. Trench and his last work on the biology and diversity of symbiotic dinoflagellates." And what I would like to try to take away from today's message is that there are indeed opportunities for anyone, no matter who you are, no matter what you look like, to make significant contributions within the field of marine biology and marine science. I'm very excited to also follow within this field of marine microbiology, and hopefully I can continue to share more about my interests and passions within this field. Thank you so much to the BIMS community and the founders for allowing me to talk a little bit about coral microbiology and some of the history associated with it. If you would like to learn more about my research and the lab that I'm doing here at Penn State, you can feel free to visit our lab website. And you can also follow me on any of my social media platforms at Marine Sci for Life. Please don't forget to share, like, and subscribe across any of the Black and Marine Science social media platforms. And I look forward to talking with you again soon. Cheers.